So the last major stop in the DNA subway blue line will be analyzing your sequences. First, you'll use a tool called BLASTn to find DNA sequences that closely match your barcode. It's sort of like a Google search for DNA sequences. Once you find those closely matching DNA sequences, you'll compare them using two different analyses. One called a muscle alignment that'll align all the sequences and see how they compare, and the other be building phylogenetic trees to compare your sequences and look at their evolutionary relationships to one another. The first step will be blasting your DNA sequences. BLAST stands for Nucleotide Basic Local Alignment Search Tool. What BLAST does is it searches for your consensus sequence against a national database of known sequences called GenBank. It's essentially a Google search for DNA sequences. To run BLAST, it's pretty simple. Just click each sample you want to blast from the drop-down in the BLAST tool, and then hit the BLAST button. Once you hit that button, it'll take a few seconds, and eventually you'll see a screen that pops up that looks like this. Then all you have to do is hit this little square. Once you hit that square, you'll have the BLAST results pop up that looks something like this. So this is what your BLAST results will look like. Here's the BLAST results for Jessica's unknown. For each sample, you'll have a list of the database hits ranked by what's called bit score. The bit score is calculated based on the alignment length and the mismatches, which you can see listed on the right side of the table. Alignment length is just the length of the sequence used to make the match, and mismatches is just the number of mismatch bases between the query or Jessica's DNA sequence or consensus sequence and each of the hits. So for example, that first hit, Pluteus ongui, with the bit score of 1061, had 588 base pairs in its alignment, so 588 base pairs matched between that hit and Jessica's consensus sequence, and there were 0 mismatches. You can also see this E value or expected value. This is the likelihood that the match or the hit that BLAST made between our query and that sequence happened by chance. So the lower this value is, the better it is. And typically these values are way, way, way below 0, which means it's not by chance that the hit matched your consensus sequence. A few quick rules of thumb regarding BLAST hits. Number one, if the number of mismatches divided by the alignment length is greater than 1%, this often means that the match is a different species. Though this is really highly variable across taxonomic groups and hotly debated. This is just a sort of loose rule of thumb to use for this course. Also, if your alignment length is less than 400 base pairs, your data often are not reliable, at least to ID to the species level. Though once again, this is highly variable depending on the primers you use, the region that you're amplifying, the taxonomic groups that you're comparing, and also hotly debated. This is just, once again, a general rule of thumb to use when you're IDing your sample for this course. If you click on an individual hit's species name or the link for a species hit, like here, Pluteus ongui, you can see some photos, some other taxonomic information that'll pop up. From here, if you want to do a little bit more research on the individual hits, you can highlight the species name and then just right-click on it and search Google for the genus and species name. And oftentimes you can get good taxonomic information from iNaturalist or Wikipedia to help flesh out the information that you need to fill in your lab notebook. I've found that asking ChatGPT can also sometimes be useful to get this taxonomic information. So what do you do with all of these hits? Eventually what you're going to need to do is select particular hits to add to your project for downstream analyses. So for example, looking at the BLAST hits from Jessica's unknown, typically what you're going to want to do is add the top 2 to 3 hits that are ID'd to the genus and species level. And this can be true even if it's the same genus and species for those top 2 to 3 hits. So for example, for Jessica, I'm going to select these top 2 Pluteus ongoi hits. They have the same bit score and they are ID'd to the genus Pluteus and species ongoi level. In addition, you want to add the remaining 2 to 3 taxonomically unique hits as rated by bit score. So top 2 to 3 is rated by bit score that have a different genus or species name. So for example, this is Pluteus petacetus, so it's a different species in the same genus, so I'll select that one. Another important detail to take note of when you're selecting hits to add to your project is you don't want to add samples that aren't ID'd to the genus or species level. So I wouldn't add this hit that says Pluteus spa or the hit below it that says uncultured fungus, because those are not going to be useful to help ID your specimen. Once you've selected sequences to add to your project from your BLAST search, the next step is what's called a muscle alignment. Muscle stands for multiple sequence comparison by log expectation. Essentially what a muscle alignment does is it takes all of the sequences that you've selected to add to your project and it aligns them or stacks them up on top of each other to compare them, and from there you can impute potential evolutionary relationships and relatedness between the sequences. So the first step in creating a muscle alignment is to add reference data that taxonomically match your samples and primers, but are not necessarily close BLAST hits. This allows you to select downstream what's called an outgroup, and I'll explain that to you in a little bit more detail very shortly. So to add reference data, what you're going to do is click the Add Reference Data, and then you'll have several choices of reference data to choose from. COI for animals, RBCL for plants, 16S for bacteria, and then ITS for fungi. So since we're analyzing fungi right now, we're going to select reference fungi from the ITS group. Once you've added your reference data, then you can select sequences to align. And this will essentially be all the BLAST hits that you selected from the previous activity, your samples in an individual outgroup. So this is how you'll do that. So this is going through right now, selecting all of your BLAST hits that you selected from the previous activity to add to the project. Then your consensus sequences of your unknown samples that you collected, and then an individual outgroup. And in this case, we're just going to select smut fungus, and then hit the muscle button. And it'll take a few minutes to analyze. After about a minute, you'll get an output that looks like this. You're going to want to trim the ends off the alignment, so go ahead and click the Auto Trim button, and you should get a trimmed muscle alignment that looks similar to this. Here's your trimmed muscle alignment. Each row in this muscle alignment represents the DNA sequence of your samples or an individual hit from your BLAST search. The labels to the left of the alignment identify each sample. The numbers above the alignment at intervals of about 100 represent the base pair position in the sequence. The light gray areas in each column represent base pairs that match in each sequence. The colored areas represent polymorphisms, or locations where the base pairs are different across sequences. Each color is representative of a specific nucleotide. A's are green, T's are red, C's are blues, G's are black. The top row that says sequence conservation represents how often there are differences in the nucleotides at that position across all the samples you're comparing. The more white you see, the more often we see a nucleotide that is different from the most common nucleotide at that position. The second row down that says sequence variation shows you all the different nucleotides that are present at that position in the sequence across all the samples that are being compared. The third row down, labeled consensus, shows you the most common nucleotide at each position in the sequence. For instance, if you align 5 sequences and the nucleotides at position 20 are A, T, A, and G, then the consensus sequence should have an A at position 20 because A is the most common nucleotide at that position. All sequences are unknown, reference, and close matches are being compared to each other. The consensus row doesn't really need to be understood in detail, but it represents the nucleotide that matches in all sequences where a polymorphism or colored line is not shown. Lastly, dark gray areas, if you see them in your alignment, represent gaps or spots where the sequence is missing from a particular sequence in the alignment. These gaps can be created by the insertion or deletion of large chunks of DNA in some but not all of the species being compared. If you hit the ATCG blue button in the upper right hand corner, it'll zoom in on the muscle alignment so you can actually see the individual nucleotides labeled by the letter. So go ahead and click that in your DNA subway screen and then answer these questions. Go ahead and hit pause until you've got the answers. Okay, so what is the maximum number of polymorphisms possible at any one site in the barcode? Well, the maximum possible number is, of course, 4. This is the maximum number of individual nucleotides that can be at any one site. It can either be A, T, G, or C. What is the most common base across all the sequences at site 5 is indicated by the arrow in the upper left corner there, site 5. The answer is A, as indicated by the consensus sequence. A is the most common letter if you compare what letter is at that point across all of the sequences being compared. And what does tray, or sample number 4, have at site 5? The answer indicated by what's in that blue square is once again A. And that's because tray doesn't show a letter there because he matches the consensus nucleotide there, which once again is A. If you don't see a letter there, that means tray, or whatever the sample is, has the most common nucleotide, or the same as what's listed in the consensus sequence. You're likely asking yourself, okay, so what do I actually need to pull out of this muscle alignment? The two big questions that you'll need to answer from this muscle alignment are, number 2, which of all the taxa that are lined up is most similar to your particular unknowns? So in this example, let's say, which of these is most similar to Trey's unknown? And you'll also need to identify which taxon or which individual sort of sequence line is least similar to all of the unknowns. So if we look at Trey's sample, it's tough to say which one is the most similar, whether it's Hymenopellis incognita, Limnospora, Megalospora, radicata, or orientalis. Likewise, it's very difficult to say which taxon is the least similar across all of our unknowns. So a really useful tool to help tease this apart in a quantifiable way is the sequence similarity table. So let's look at that on the next slide. So once you click on the sequence similarity table, you should see something like this. This is a % sequence similarity matrix. It's a table that does a cross comparison of every single sequence that you're looking at in your muscle alignment. Sequences labeled 1 through 24. So for example, we know that Trey's sample is sample number 4. And so we can look at the highlighted area here and follow that row the whole way across to see which % sequence similarity is the highest between Trey's sample and the samples that we're comparing him to. And what you can see is 96.77 is the highest number. So samples number 5 and 6 are 96.77% similar to Trey's sample. They have the highest % sequence similarity in that muscle alignment. And so those samples, Hymenopellis, are both Hymenopellis limnospora. And so they have the highest sequence similarity to Trey's sample. We can also begin to decipher which taxon is the least similar to all of our unknowns. Can you hit pause and figure it out on your own? If you said sample 1, you're correct. If you look at the % similarity of sample 1 to all the other samples, you'll find that sample 1, the smut, is the least similar, about 55 to 67% similar across all our groups. So sample 1 will be our out group. And apologies that it says baker's yeast there, it should say smut. Smut will be the out group. Now that you've aligned and compared your sequences in the muscle alignment, you can begin to build phylogenetic trees from those sequences. Phylogenetic trees are a graphical representation of relationships among taxonomic groups. DNA Subway uses a tree-building algorithm called PHILIP, or phylogenetic inference package, to attempt to reconstruct the order in which sequence mutations accumulated as different lineages diverged from a common ancestor. This software will create neighbor joining, or NJ, and maximum likelihood, or ML trees, by analyzing the similarities and differences among the DNA sequences you ask it to compare. These two types of trees are constructed using different methods, with NJ trees tending to be a bit simpler to interpret at the cost of being a little less accurate relative to the ML trees. There's two major things that you'll need to provide the software if you want to create a phylogenetic tree. Number one is you'll need to input known barcodes of closely related species. In other words, your BLAST hits. And then you'll also need to have known barcodes of a more distantly related species for comparison or rooting the tree. That's called the out group. This helps root your tree by determining which nucleotide changes are more ancestral versus derived. Let's make an ML tree first. The ML tree will automatically analyze all the taxa that were present in the muscle alignment you previously constructed. All you have to do is select an out group to root the tree. In this case, we've selected smut fungus. How did we know to use the smut fungus as the out group? Hit pause until you know the answer. If you said because it was the least similar to all the other taxonomic groups that we looked at in our percent similarity matrix, you are correct. This is the maximum likelihood tree output. This algorithm fits mutations to a tree to produce the most parsimonious tree. In other words, the one that requires the fewest number of mutations to explain the sequence data, aka the shortest branch lengths. Internal nodes on this tree, represented by the big blue dots, represent a common ancestor shared by taxa on the branch tips. Horizontal lines represent sequence differences, aka evolutionary change. The longer the horizontal branch is, the larger the evolutionary distance, aka the less related those taxa are. For example, you can see that Mike's sample has a very short branch off the Pluteus cervinus and Petacetus clade, and therefore is closely related to those two species. In contrast, the smut fungus down at the bottom, that outgroup has a very long branch, as the outgroup should, demonstrating its distant relation to all the other taxa. Next, let's make an NJ tree. Same as the ML, the software will automatically analyze all the taxa that were present in the muscle alignment you previously constructed. All you have to do is select an outgroup to root the tree and hit select. We'll use the same outgroup as before, as the smut fungus is still the least similar taxon from the percent similarity matrix in the muscle alignment. Your output should look something like this. The neighbor joining algorithm builds a tree from the bottom up by comparing the evolutionary distance between pairs of DNA sequences. Sequences with best matching sequences are linked as neighbors that share common nodes in the tree. Because the branch distances are produced in a pairwise manner, neighbor joining does not optimize branch length and tree parsimony. The numbers you see on the tree typically represent bootstrap values. These values indicate the confidence level in a particular branch of the tree, reflecting how frequently percentage-wise that branch appears in multiple trees generated from resampling your dataset over and over again. Higher bootstrap values suggest a more reliable branch. For example, a bootstrap value of 95 means that 95% of the resampled trees contained that specific branch in that specific location, indicating a high level of confidence in that branch's existence in the quote-unquote true tree, if such a thing exists. A common rule of thumb is that values above 70 or 80% in bootstrap are generally considered well supported, though this can vary depending on the specific study and data set that you're looking at.